Good evening, and thank you for joining us for the 16th Florida Keys Mosquito Control District Oxitec Public Educational Webinar. Tonight, we'll be looking at takeaways from our project last year in 2021 and our plans for this year in 2022. With us tonight, we have Andrea Leal, the Executive Director of the Florida Keys Mosquito Control District, Nathan Rose, Oxitec's Head of Regulatory Affairs, and I'm Meredith Vincem, a Florida native and Oxitec's Head of Public Affairs. So this webinar is the 16th in our series, and we are relaunching them now to continue once a month through the mosquito season and possibly beyond. This is to provide a forum for especially residents of the Florida Keys to ask questions and get them answered. The webinars are open to everyone. They are recorded and made available for everyone after the event. We post them on Oxitec's YouTube channel, the project website, keysmosquitoproject.com, and the oxitec.com Florida webpage. We're going to spend the first half of the webinar doing the presentation, and then the second half, so the last 30 minutes, we'll have a Q&A. We will answer all of the questions received. We may batch some similar questions together. And if we run out of time, we'll accept questions in writing and we'll publish them afterwards. Just for you all in the audience, you will not be identified when you submit your questions. We're asking you to use the Q&A box. We will see your questions, the audience will not. If you can please identify, if you're a Florida Keys resident, which key you live on, we will give priority to residents of the Florida Keys. So our agenda is takeaways from the 2021 project. What did we achieve? We'll review the plans for 2022, and then we'll do our Q&A. Now over to Andrea.
Thanks, Meredith. So I think we kind of like to start by talking about why we're looking at this, what is going on in the Florida Keys, and really what's up with the mosquito itself. And this mosquito in particular is called Aedes aegypti, and it makes up about 4% of our total mosquito collections, but is actually responsible for 100% of the diseases that we've seen here recently. The main issue that we're seeing is dengue fever. When we did have an outbreak in 2009 and 2010 in the Key West area, and then again had just over 70 cases in the Key Largo area in 2020. The mosquito is very difficult to control. Its behavior likes to live around people, in houses, under houses. So it's very difficult for the products that we currently have to control this mosquito. On top of that, we're also seeing an increase in resistance to a number of our adulticide products that we use for mosquito control. So at the end of it, it really is, we're looking for new tools to put in our toolbox to help us control this particular mosquito. There is no silver bullet. And, we're just really hopeful that we are finding something that we can integrate with the rest of our control methods. And again, here in the Florida Keys, being surrounded by a national marine sanctuary, it's very important that, we're looking at products that don't have environmental impacts. And that is definitely something we take into consideration when we're looking at new products. So that's really what brought us to looking at the Oxitec mosquito. And, hopefully with what we saw this last year and hopefully with getting more information this upcoming year, we can find another tool that we can integrate. Nathan?
Great, thanks, Andrea. So what I thought we'd do is just start by a brief introduction to what Oxitec's mosquito is, for those who are not familiar with this project. and then we'll talk a little bit about what we set out to do in 2021 and what we were able to achieve. So Oxitec's Aedes aegypti male mosquitoes released by Oxitec go out and mate with invasive female mosquitoes of the same species. And only the male offspring of these encounters actually survive. So we release male mosquitoes only. Males don't bite, only females are able to bite. And the mosquitoes that we release give targeted control of just the single species. So you're not having impacts on any other species because this happens via mating. The mosquitoes that we release are safe, they're non-toxic, they're non-allergenic. We have a strong track record of proving these to be effective in Brazil. And we saw some great results in the field in Florida last year as well. And our mosquitoes as well are male-only releases. They're traceable in the field because we have a fluorescent marker, which is part of these mosquitoes. And importantly, they're also self-limiting in the environment. So once releases stop, these mosquitoes actually don't persist long-term in the wild. They will disappear within a few weeks or months. And that's exactly what we're seeing in Florida right now too. So briefly, just to talk about what we set out to do. Oxitec, when we release these mosquitoes, we have 3 different kinds of field projects that we might carry out. The smallest scale is what we call a regulatory pilot. And this is where we do small studies with high statistical power by making sure that we're repeating the studies that we're doing. We have protocols that we agree together with the regulators. And this is really to look at the biology of the mosquito and also its efficacy as a means of killing the next generation of the female mosquito. And that's what we set out to do last year, and that's what we focused on. Following on from regulatory pilots, something that we have done in Brazil as a company is to do larger scale demonstration pilots where we actually release these mosquitoes over much larger areas of a city, looking at suppression, and then obviously moving towards operational deployment as well as a vector control tool over a larger area. And so, as I mentioned, last year's project was very much in the category of regulatory pilots, where we designed protocols together with the regulators to be able to answer the kind of questions that they would have when they actually assess this as a mosquito control product. So just a reminder, last year we had two major parts to this project. One was single point releases, where we released mosquitoes from single locations, and then we trapped the males and also their offspring. We also had multiple point release projects where we released these mosquitoes throughout small neighborhoods in different parts of the Florida Keys, and again, trapping the offspring. And then we had replication and comparisons with untreated areas where no mosquitoes were released. And the locations where we actually carried out these releases were determined following community engagement throughout the Florida Keys. And last year's project focused on a number of specific elements agreed with the regulators. So that was the EPA and then also the Florida state regulators, FDACs. And so we were looking at how far the males would fly, how long they're actually present in the field, the duration of the effect after releases stopped. We looked at natural, or what are called cryptic breeding sites. So these are places that these mosquitoes might go and lay their eggs in and around people's homes. And then importantly, we looked at the percentage kill of the female mosquitoes, so the next generation, and then also the percentage of the invasive population treated. These were small scale studies. They were not designed to measure suppression over large areas. These were limited in time and also in scope to really address these particular elements that the regulators were interested in seeing. One of the questions we often get is how you actually collect data in this kind of project. So we release mosquitoes from the boxes where we basically put mosquito eggs into the box, we add diet and we add water. And after kind of 10 to 14 days, we have the male-only mosquitoes coming out of those boxes. What we then do is we have a couple of different types of mosquito traps that we placed throughout the project areas to be able to assess what was happening. So one of these is called an overtrap or an egg collection trap. And these are just small plastic cups that have some water in them, and they have an egg laying surface. And this is a perfect environment for an Aedes aegypti female to go and lay her eggs. And then what we would do is we would take these eggs back into the lab where we could hatch them, and then we could see whether those eggs were actually from one of our males that we released by looking at the fluorescence, or we could see whether the eggs were from a mating between a wild invasive female and a wild male mosquito. Second type of assessment is using adult traps, and this allows us to actually capture adult mosquitoes. And this, what we can do then is we can look at the ratio, for example, between the number of Oxitec mosquitoes and the number of wild mosquitoes that are actually out there. And then finally, the lab-based assessment of all of the mosquitoes that comes from these is really important. And there we use stereo microscopes. We can screen for fluorescence of the mosquitoes which are coming in. All right, so just a reminder for those who weren't following this project closely last year, these releases occurred in selected locations in the middle and lower keys. So this was on Vaca Key, Ramrod, Cudjoe, Little Torch, and Big Coppet keys. And we had untreated control sites in Key Colony Beach, in Little Torch Key, and in Summerland Key. And these releases began in late April 2021. And we had our last releases happening, I think, just before Thanksgiving in November 2021. Unfortunately, we also had two significant interruptions to this project from tropical storms Elsa and Fred that came very close to the keys in July and August. And so during those storms, following the protocols which were approved by the regulators, we paused our releases during those storms. And then afterwards, we were able to resume the project. So this is a brief overview, and then I'll go into some more detail on each of these specific points after that. But really, we had quite a number of key performance outcomes that we were hoping to hit, and we were able to hit all of those in this trial. So we were able to measure the self-limiting genes effectiveness in the field. We were able to look at dose rates that are suitable for use of this as a mosquito control tool. We looked at male performance using a number of different measures. We looked at box dosing to understand how our dosing related to the number of wild mosquitoes that were out there. We looked at male mating effectiveness. We looked at the ability of females mated by our males to actually access those cryptic breeding sites, which was a really nice outcome. And also we confirmed that we did not have any females released throughout the project. So I'll start off by looking at the dispersal of the mosquitoes which were released. And so you'll remember that I mentioned that we had these single point releases where we would release mosquitoes from a single location. And in doing this, we confirmed that the flight range of Oxitec's male mosquitoes was the same as that of wild Aedes aegypti males worldwide, basically. These mosquitoes typically travel about 50 meters during their lifetime. And that's exactly what we saw in this project. So in Project A, where we had these single release locations, we had the mosquito coverage, so the male mosquitoes that came out of this box concentrated within a 50-meter circle, equating to about 1 hectare around those release locations. And that was as expected, and it's what we've seen in other locations as well. We also had mosquito releases throughout these small neighborhoods in Project B, and there we saw effective mosquito coverage throughout those release neighborhoods. So we spaced these boxes based on the kind of coverage that we saw in Project A. And then we measured coverage throughout those neighborhoods with traps throughout those neighborhoods. And we saw that that kind of spacing was very effective in giving us a good mosquito presence, good male mosquito presence throughout those neighborhoods. So this was a great outcome. We were really happy to see this. We're not particularly surprised because this is definitely what we know about this mosquito's biology from other locations. But it was great to see this confirmed in the field in Florida. And the other thing just to say is that we were monitoring up to 400 meters from those central release locations in Project A. And we didn't see any of the released male mosquitoes, the ones that came out of the box, we didn't see any of those at that 400-meter radius. And it really just reinforces previous data again about how far Aedes aegypti can actually move. The next really important part of this project was looking at effective mating by the male mosquitoes that we released, and also what happened to the female offspring of these matings. And here we confirmed that the larvicidal efficacy of our mosquitoes was 100% throughout the project. So throughout the entire field season, we were taking eggs from those ovitraps, bringing them back into the lab, hatching them. We screened over 22,000 Aedes aegypti larvae for fluorescence. And then what we did is any larva which was fluorescent at the earliest stage from after these eggs hatch, we would then track that through to adulthood. And we saw that every single fluorescent female larva died before it reached adulthood. So again, this is what we've expected, and it's what we've seen in the field in Brazil and in the lab as well. And you can see on the right-hand side there's some pictures of these fluorescent larvae under a microscope, and you can see the actual results from each of the different project areas. So in Project A sites and in Project B sites, we had 100% larvicidal efficacy. 3 of the Project A sites did not detect any progeny from our mosquitoes, but this was not too surprising because those sites actually ended up having a very low presence of any wild Aedes aegypti throughout the season, even though before the season we'd been monitoring there and we had seen enough presence there to think that it was worth carrying on the projects there. We actually saw that the presence throughout the season was pretty low in those sites, and so we didn't see any fluorescent larvae in those sites. But in the other sites, we saw significant numbers of fluorescent larvae, and as mentioned, all of these that were female died before they reached adulthood. We also, in accordance with the protocol which EPA agreed with Oxitec, we carried out regular PCR, so that is genotyping based confirmation, also confirming that the screening accuracy, so our ability to tell whether a larva was fluorescent or not, was highly accurate, and we didn't have a single sample that was misscreened throughout the entire season. So this is great. It reflects the complete penetrance of this conditional female-specific self-limiting gene, which causes lethality in all of the female offspring from the released Oxitec male mosquitoes. I also mentioned that we were really pleased to see progeny of our male mosquitoes found in cryptic breeding sites. So in Project B, where we had these neighborhood-wide releases, we were having the technicians who are out in the field actually monitor natural breeding sites, and if they found any fluorescent larvae in those sites, we could identify what those were. And so we actually found fluorescent larvae in a number of different natural breeding sites, things like flower pots, plant trivets, garbage can lids, buckets, tires, toilets, even jet skis, soft drink cans, and so on. Obviously, the best thing to do is to get rid of any of these kind of breeding sites that you might have in your backyard. It's not great to have any of these kind of breeding sites for Aedes aegypti or any other mosquito. But where we did find these, we were also able to confirm the presence of fluorescent progeny from our mosquitoes. And as Andrea said at the start, this is really helpful because one of the challenges with traditional mosquito control tools is actually being able to access all of these hidden or cryptic breeding sites effectively. So if Oxitec's male mosquitoes are able to find the invasive females before they lay their eggs in these cryptic sites, that's a way of actually controlling this mosquito in the places that are hard to reach. Then following the end of releases, we, in accordance with the protocols, have been monitoring all of the areas where we released mosquitoes until we see the complete disappearance of any Oxitec fluorescent mosquitoes from those locations. And once we see no more mosquitoes, we then have to continue monitoring until we have 10 weeks with no fluorescent mosquitoes detected. And the reason for that 10-week period is that that equates to at least 2 full life cycle generations of the Aedes aegypti mosquito. So it takes about a month for that mosquito to complete a life cycle. So we have completed post-release monitoring in all but 3 of the release sites as of last week, the 29th of March. So I'm only talking about data up till that point. So the sites which were low-dose Project A sites, those were completed, I think, pretty much 10 weeks after the storms, because we stopped releasing those sites when the storms came. The higher dose Project A sites, which I'm showing here on this particular graph, the last time we saw fluorescent larvae from any of those sites was in the 3rd week of 2022. So releases stopped around about week 47, 48 of 2021. We've then been monitoring since that time. And sites A1 and A2 have not had any fluorescent males now for at least 10 weeks, and sites A15, we're almost at that point. And then in the Project B sites, so the last time we had any capture in a Project B site was in week 8 of 2022. So that was, I think, sometime in February. And we will continue to monitor until we hit that 10-week period with no fluorescent mosquitoes in each of those sites. So this is, again, pretty much what we expected. In Brazil, when we've released these mosquitoes, we've seen a bit of presence of the fluorescence for a few weeks or a couple of months after the end of releases, and that's exactly what we're seeing here. and then we start to see that that completely disappears from the environment. So this really just confirms the self-limiting nature of the genes. They're not going to persist long-term in the environment. Finally, I just wanted to touch on project compliance. So there were a lot of conditions placed on this project by the regulators. So that was both the federal regulator, EPA, and also the state regulator, FDACS. And I just wanted to briefly update on these. So we had no adverse effects on human health or the environment observed throughout this project. No adult female mosquitoes carrying the Oxitec transgenes were detected throughout the project. We also had caged release boxes throughout this project, partly to help us measure the release numbers, which is something the regulators want, but also it enabled us to confirm that no female Oxitec mosquitoes were released from these boxes. We actually had 2 project audits from the FDACS Bureau of Inspection and Incident Response. So that happened in June and in September 2021. And we had full compliance of the releases observed in each audit. And as I've just mentioned, post-release monitoring has been initiated. It's been completed in most sites. And once we have completed all of that, we will notify the regulators as well. And then finally, we had no Oxitec mosquitoes released within 500 meters of sewage treatment facilities or commercial citrus groves, again, as required by the permit. So I'm going to hand over to Andrea now to talk about what we're planning for 2022.
Thanks, Nathan. And as a reminder to those on the webinar, you can go ahead and start entering any of your questions at any time in the Q&A. So don't be shy. We're here to answer all of those questions. So as far as upcoming plans, we are hoping to follow up on all of that information that Nathan just shared with you as far as what we learned in this last year. We want to continue looking for more data collection on dispersal, how long are those mosquitoes living, how well are they mating with our wild females, especially over small areas and in single home areas. And really just collecting more data to support a pathway to potential commercial registration. We're looking forward to collecting as much information as we can here at Mosquito Control. It's important for us to really see if this is going to work as something that we can supplement with the rest of our control methods. So that's why it's important for us to continue moving forward, collecting all of this data. So where we are right now as far as regulatory approval process, the EPA did approve the extension to our current experimental use permit. So we are good until 2023 through the EPA. And that extension was granted after a public comment period and extensive scientific evaluation. So for anyone that's interested in that EPA information, the link is there and available. It's got really great information through all those risk assessments that have been done. An application has also been submitted to the Florida Department of Agriculture and Consumer Services, who is our state regulatory authority here in Florida. And they are right now pending approval, and we would anticipate having an answer from the state within the next 2 or 3 weeks. So we are hoping to have all of our approvals in place, and we'll be able to begin releases in the spring of this year. What we're looking at doing is pretty similar to last year, especially when we're looking at Trial B. And it's actually really just like Project B from last year, only in larger areas. And this is going to help us get a good idea of how much of that population are we treating with these releases, how long are those mosquitoes lasting, the presence in cryptic breeding sites again. And this is one where we are hoping to see if we can get local population suppression. It's important for us to know what are the effects on our local population. And that Project B is going to hopefully be able to start answering some of those questions for us here locally. Trial D, which is going to be a single household study. So it'll be a single point release and then traps all throughout that particular site. And it'll give us a good idea to know what effect just one single point release is having and at that household level. Trial E is a mark release recapture, and this is really looking at dispersal. Again, we did get great information from our previous studies in 2021, but it's important to continue looking and making sure that these mosquitoes are behaving in the ways that we think they are. So this mark release recapture will really give us that information on dispersal and the longevity, how long that they are staying in the environment. So right now we are very heavily involved in community engagement. We've got a number of potential sites that we're looking at but have not finalized those sites and won't until we have full regulatory approvals in place. But we have been going door to door in sites that we're interested in moving forward with, just talking to people, getting a good gauge on participation. And I will say that in all of the communities that we've gone into, we've been very welcomed and have found that more often than not, folks want to participate, which has been a really great thing. So I think that, we're on the right track here. We've also started pre-release monitoring in a number of these sites again, because we are hopeful to get those regulatory approvals in place within the next few weeks. And then hopefully we can start mosquito releases soon after. Project B that I described, which is basically the neighborhood-wide releases, is going to be the first project of this year to be initiated. That's the one that you really need to start our releases before high season, and we want to continue those releases throughout all of season to get a better understanding of the effects on our local mosquito population. And then projects D and E will follow a little later in mosquito season. But we'll be sure to put out press releases, make sure everybody knows what's going on as far as all these projects and the timelines and the locations. We are going to identify by key where the release locations are going to be, similar to last year. And all of that information can be found at keysmosquitoproject.com. So I would say, just keep taking a look at that website. We'll have updated information throughout all of the season to let everybody know what's going on. And now over to you, Meredith.
Thank you, Andrea. Everybody, we'll start with the Q&A. We've received fewer questions than usual, so just want to encourage you all, don't be shy. I mean, anything you're curious about, someone else is probably curious about too, and the audience will not see your name or your question, just we'll see it and we will answer it anonymously, but we will give priority to Keys residents if you can tell us which key you live on. Okay. I'm going to take this first question. So why aren't there any peer-reviewed publications available about your project last year? And I think probably either Nathan or Andrea could take that. I don't know if there's a volunteer.
I'm happy to take that if that's, yeah. So this project is still ongoing. As we've mentioned here, we completed the releases in November, but we were still in this post-release monitoring period in some of these sites. And we also think that it's important that we continue, as Andrea mentioned, this year to really strengthen and supplement the data which we were able to collect last year. So I think once this project is completed, we would then be in a position where we could actually put a paper together. And then obviously any peer review publication depends on a journal editor wanting to send that paper for review and then also actually accept a paper following review. So I think that's the process that we will be following here.
Thank you, Nathan. The next question, I'll ask Andrea to answer it, and then Nathan, if you have anything to add, it's welcome. The question is, did the storms last year affect the evaluation of the project?
So the storms last year definitely affected collecting of that information in a contiguous manner, right? So the protocol, according to the EPA and FDACS last year, stated that, should a named storm come close to the Florida Keys, that all of those release boxes and traps needed to be removed prior to that. So, at that point, with everything pulled out of the field, by the time it all gets set back up, we're looking at about a 2 to 3 week lag time in releases, which really affects the data that you're trying to collect. So, we're hoping that we can make improvements to that in the upcoming year as well. But, we did our best. And I think as a scientist or anybody that's been involved in studies, field studies, nothing ever goes completely as you hope it does, which is why we do these repetitious studies to make sure that, we can collect all that information that we need.
And just to add, we really appreciated the partnership from FKMCD in helping us to get those projects back on track again after those storms. And despite that, we were able to have a really good field season, as I hope you've seen in what we've talked about this evening.
Thank you both. This next question, Nathan, I will send to you. And the question is, have you considered using your technology for iguanas? Would that be possible?
No, honestly speaking, I don't think we have. I think it would be pretty challenging to do something like this and anything more complicated than an insect. But yeah, maybe Andrea, I don't know if you have any thoughts on iguana control in the Florida Keys. You probably know more than I do about that.
I know that they're an issue, but definitely not my specialty. So we'll leave that to the iguana experts.
I think the thought of releasing more iguanas to deal with iguanas would be quite a challenge.
Okay, thank you both. This next question, Andrea, is definitely for you. It is, how did FKMCD feel about the project?
Well, like I said, field studies are difficult, but I think, working very closely together, it was a lot that we learned. There were a lot of things that we learned. There's a lot of monitoring that went into it, a lot of identification. But, together, I feel like working with Oxitec and our district, we did a really great job in getting these pilot studies completed and some really good information to really build off of in the future. Yeah, I do appreciate the partnership, and I know that, we all worked very hard to get all this information together.
Thank you, Andrea. The next question is also for you, Andrea. The question is, did you see changes or effects on other mosquito species during your releases?
So we didn't observe any changes in the collections that we had with, as far as adult and larval collections that were done throughout the project. But there were some pretty cool things that were found. Speaking as a bug nerd, we did find Aedes bahamensis, which has not been previously found in the Florida Keys. It has been found in Miami-Dade and in Broward County, but this was really the first time that we've seen it in these areas of the Florida Keys. So, that was a really cool thing. And, what it means, obviously at this point, it was only a couple of species. We're not seeing, any new movement really coming in and establishing large populations. But it just kind of goes to show you that when you're really looking hard, you can find things that maybe you've never seen before. So it was just kind of a cool thing, and is actually out for publication right now.
Great. Thanks, Andrea. Okay, the next question, Nathan, this is for you. It's, how do you tell the difference between your mosquitoes and the invasive Aedes aegypti mosquitoes?
Yeah, that's a good question. So the Oxitec mosquitoes have a fluorescent marker gene in them, and their offspring also inherit that fluorescent marker gene. So that means when we look at them under a microscope, we can actually tell whether it's an Oxitec mosquito or one of the wild invasive ones. And then the other thing I mentioned is we can confirm that using genetic methods. So something called PCR, allows us to confirm whether that mosquito has been correctly identified using the fluorescence screening or not. And that's something we did throughout the project. It was part of the protocol requirements, and we confirmed that the fluorescence screening that we were doing on all those samples that came in from those overtraps was accurate. So there's definitely those two different routes that you can use to tell whether it's an Oxitec mosquito or a wild one.
Thank you, Nathan. The next question is for you also. The question is there any consideration about expanding this project to the mainland? I live in Miami-Dade, so not exactly the Keys, but right there.
So at this point, our focus is really just on collecting the data that we need to go back to the regulators to help them to assess whether this is an effective mosquito control tool. And so the way we're doing that is in partnership with Florida Keys Mosquito Control District. And then we're also, as some people might be aware, planning a project in California. But it's really just those two areas at the moment, focused on collecting the data which the regulators will want to see. Once the regulators have assessed that, if they were to grant a commercial registration for this, then there would be the possibility for this to be used more widely by other mosquito control authorities or possibly even by individual consumers. So Oxitec is already using this commercially in Brazil, and these mosquitoes are actually available for sale to individual consumers as well. And so there's definitely scope for that in the future, but right now we're still in this pilot phase.
Great. Thank you, Nathan. Nathan, the next question is for you too. But first, I just wanted to say we're getting more questions now. But if you do live in the Keys, please say where you live in the Keys, and we will prioritize those. This next question is, what is the exact technology Oxitec uses to ensure no females hatch from the mosquito boxes?
It's a good question. So it's actually very simple. What we do is we put some of our mosquito boxes into cages that allow us to basically trap all of the mosquitoes that are coming out of those boxes. And then what we can do is we can look at those mosquitoes, and we confirmed throughout the project that all of those mosquitoes coming out of the boxes were Oxitec male mosquitoes. This is also following extensive quality control that happens before we even ship any mosquito eggs to Florida. So we have very stringent quality control processes that happen in the lab in the UK, where we also confirm that the mosquitoes are functioning exactly as expected. Then we can ship the eggs to Florida with confidence that they've already undergone this very stringent quality control. Then we're also checking in the field using cages around some of these boxes. So it's pretty stringent, and we're pretty confident, again, that we're not seeing any release of any Oxitec female mosquitoes.
Thank you, Nathan. Nathan, the next question is also for you, but Andrea, if you have anything to add to this one, it's welcome. The question is, will they, and they is FKMCD Oxitec, publish that information for public review?
So I think we've kind of answered this question already, in the sense that this project is still ongoing. And the hope would be that once we've completed this project, we have a really good data set that we can obviously submit to regulators, but also that we can put together into a peer-reviewed publication. So I think that, for me, would be the ideal way to actually make this available. But Andrea, please feel free to comment as well.
Yeah, exactly. I mean, with the interruptions that we saw this last year, it's important that we gather more data. And of course, at the end of all of this, we would love to see a peer-reviewed publication that comes out from all the work that's been done.
Great. Thank you both. The next question, Nathan, this is for you. I'm not sure, I get what I understand what they're asking. I'm not sure it's a complete question, but is there any reason not to believe the wild type females, so talking about the invasive females, will not eventually discriminate against the Oxitec mosquitoes?
Okay, that's a really interesting question. So I guess the question is there any possibility that wild females would suddenly at some point realize that if they mate with an Oxitec male mosquito, their offspring are going to die? No, I don't think there's any possibility of that happening. And the reason I say that is that Oxitec has released well over 1 billion of its mosquitoes in different parts of the world before this project in Florida, mostly in Brazil, but also in some other countries. And we've never seen any evidence of the wild females discriminating against Oxitec males. And one of the really key ways that we know that is that we see effective population suppression. So if you're seeing suppression of the population, that means that the wild females are mating with the males that we release, and we see their offspring dying, and that has the effect of suppressing those populations. So I don't have any concerns. The other thing I would say is that the idea of releasing effectively sterilized males, obviously in our case they're not sterile because the male offspring survive, but the idea of releasing sterilized males using, for example, irradiation to sterilize males has been around for probably 60 or 70 years. It's something called the sterile insect technique. And that's been done with a whole lot of different insect species. And I think there's only one instance that's been found of any possible discrimination against those released males. And in that case, they did some follow-up studies and they tracked it back and they found that it was actually just because the males that were released were really sick and were not effectively mating. So the females were not choosing them because they were visibly hobbling around or whatever the case was. So very extensive track record, both on the sterile insect technique side and then also with Oxitec's releases, really does show that there is no concern about wild females discriminating against the released males.
Thank you, Nathan. The next question I'll answer, but if anybody else wants to weigh in, it's welcome. The question is the public support for the project any different since 2021? I would say it's similar to last year. There's a lot of interest, a lot of support. If anything, the interest and support has grown. But yeah, I would say it's similar to last year. And I think you all who live in the Keys and are watching know that on our project website, keysmosquitoproject.com, we have a Join the Project tab where you can go and sign up to volunteer to host a mosquito box or a trap or to receive our project updates. We have established a pretty extensive listserv for the project, and we send updates out regularly. But we do have waitlists established really throughout the Keys of hosts who want to be a part. So we're proud of that, and we're proud of the levels of interest and support.
Well, and if you don't mind, I'll jump into what we've said all along at the district is we wanted to make sure that these releases were occurring in areas where people wanted us to be. And so I think that, focusing on the areas that the referendum information was essential. And what we found is as we're focusing on those places that, as Meredith said, more often than not, we're getting, more folks that want to sign up to either host a release box or host a trap. So, I am very grateful for the community support that we've seen.
Thanks, Andrea. The next question, Nathan, this is best for you. The question is, were the regulators happy with the project?
It's an interesting question. So I would say yes. The regulators in Florida carried out 2 project audits, as I mentioned, in June and September last year, and they were very happy with the project compliance that they observed then. And then I guess the bigger question is, are they happy with the data? So far, yes, I think a lot of interest in what we've seen in Florida. Obviously, the data set is still not complete. And so we'll be continuing to carry out this project this year if we get regulatory approval to do so from the Florida state authorities. But certainly from the EPA perspective, I think their approval of the extension of this project is definitely a vote of confidence in how it's gone so far as well. So yeah, I think the answer is yes.
Thank you, Nathan. The next question really could be answered by either of you. It's how many homeowners? Well, this says volunteered to be a part of the project. I don't even know if we know the total number. Maybe talk about the number of households who were ultimately actively involved. We have that. We definitely know how many households hosted.
Yeah, I think we had something like 570 households that helped to host either release boxes or some of the project traps, the adult traps or the ovi traps last year. I don't have the exact number, but it was that kind of range. Andrea, maybe you have a better...
No, that sounds about right. Once you put all of the trap sites, adult and ovicup traps, release boxes, it was close to 600 households altogether, I believe.
Yeah, we really appreciate the participation of the community. I mean, we couldn't do this without everybody, and monitoring is a big part of this project, and that's why those numbers sound kind of high.
And just to say, we really appreciate that support as well, given the COVID pandemic and all the restrictions that placed on everyone as well. So we're very grateful for that.
Exactly. Thank you. And then Nathan, this is for you. What causes the female offspring to die?
Okay, yeah, great question. So the male mosquitoes that we release carry, as I said, 2 different genes. One of them is the fluorescent marker gene, which helps us to tell our mosquitoes apart from the wild ones. The other one is a self-limiting gene, and that gene is inherited by the male and the female offspring, but it only gets switched on properly in the female offspring. And when it does get switched on in the very early stages of those females' development as larvae, it produces a protein inside the cells of those females, which stops those cells from developing properly. And so what that does is it causes those female mosquito larvae to die before they become adult females. That protein itself is non-toxic, it's non-allergenic to any organism. It only works inside that mosquito, inside the cell nucleus of that mosquito, to stop those cells developing properly. So it's a very safe and effective way of actually getting rid of those female mosquitoes.
Okay, thank you, Nathan. The next question, Andrea, this is definitely for you. Is FKMCD avoiding treatment in Oxitec areas?
No. So that was another thing that we promised when we looked into this, is we are looking for a tool to integrate. We wanted to make sure that our control methods would continue no matter where these mosquitoes were released. So, and that's exactly what happened. If we needed to send a fog truck for salt marsh mosquitoes in one of these release areas, we would do so. So our normal control operations did continue. We just, needed to report exactly what we were doing. So if there were any effects, then that's something that we would know that had happened. So it's something, again, that we'll always look at in the future. There's a need for every single piece of mosquito control, whether it's, killing the mosquitoes as larvae or killing adult mosquitoes, or looking at, these new innovative tools. So it's just very important to have an integrated mosquito management program.
Thank you, Andrea. The next question, Nathan, I'll send this to you. Will 2022 be the last project, or will you announce more in the keys after that?
I think it's more a question for Andrea than it is for me. But I would say that the way we've set up this project in 2022, we're really hopeful that we can collect all of the data that we need to be able to give that data package to the regulators for them to be able to assess this. That's very much our plan.
Thanks, Nathan. Okay, the next question, I'll start to answer it. And Andrea, if you have anything to add, it's welcome. The question is, who paid for the 2021 trial and who will pay for 2022? Does taxpayers' money go into these studies? For Oxitec, I'll say that Oxitec is fully funding these pilots. Andrea, I don't know if you have anything to add to that.
Yeah, I can jump in. So it's important when we're doing any of these types of studies that our district personnel are involved as well. So there were no extra costs for the district, but we did have personnel that were involved in the project. Not entirely. They would just, do certain things at certain times of the project. But I can also say that that's very similar to all of the other, studies that we've been involved with, whether it was releasing Wolbachia males, when we're looking at new product trials. Those are things that, our district personnel are also heavily involved in. So I would say that Oxitec did foot all of the bill, and there was no extra funds that were used by the district for this particular project.
Thanks, Andrea. The next question is for you also, Andrea. The question is, were there any local dengue cases during the project?
So I'm knocking on wood, as always, when I talk about dengue. Our last dengue case in the Florida Keys was in August, I believe, of 2020. So we have not seen any cases since that time. But it is something that we're always concerned about, keeping an eye on, in close contact with the health department on, because, we do have the mosquito that is here, and our district is doing our best to keep that population down. But unfortunately, like I said at the very beginning, it is a very difficult mosquito to control. And, with travel opening back up, we could see introductions occurring on a normal basis, which I believe what we've seen here in South Florida in the last decade is there's a lot of travel, and there are a lot of these mosquito-borne disease introductions that are happening. So it's just important for all of us in mosquito control to remain vigilant, and why we remind you every week, every time we can, to walk around your yard and dump out any standing water that you may find, because we can all do our part. And it's important that as a community, we do our best to control this mosquito.
Thank you, Andrea. Nathan, the next question I am sending to you. I hear Oxitec are doing California projects. Will they be the same or similar? When will that detail be public?
Okay, yeah, great question. So Oxitec is planning to do similar kind of projects in California. So we are planning to work together with the Delta Mosquito and Vector Control District, which is in a city called Visalia in Tulare County in California. Again, we've been invited there by that district to work together with them. And the approval which EPA gave to Oxitec about a month ago was for the extension of this project in Florida, but also the same protocol effectively in California. So the different parts of the project that we've talked about tonight, the Project B, these neighborhood-wide releases, the single homeowner releases in Project D, and then these mark release recapture studies, Project E, that's all in the protocol for California as well, approved by EPA. In California, this also then has to go through a state approval. So right now, this is with the California Department of Pesticide Regulation, and they will do their own assessment on this, and they may request changes to those protocols, or additional monitoring, or we don't know yet. They still have to actually carry out their reviews of these protocols. So the final format that project in California would take obviously depends on the state regulators as well. And once we have more details about that, I'm sure we'll be announcing more details.
Thank you, Nathan. Nathan, the next question is also for you, but Andrea, if you have anything to add, it's welcome. The question is, were there any human or environmental health effects of impacts? Maybe that's supposed to mean or impacts from these releases in 2021?
No, simple answer.
Great.
Yeah.
Thank you. Okay. Okay, and for this year, we may not know the answer to this, but the question is, when will the 2022 project finish?
So the plan is to, as Andrea said, once we have state regulatory approval, the plan will be to start in the spring. so that we can really start at the beginning of the mosquito season. And then the other parts of the project, Project D and Project E, will kick in a little later in the mosquito season. And I think we plan to run this throughout the mosquito season in 2022. So that typically finishes around October, November time. And then obviously post-release monitoring like we did following the 2021 releases.
Great. Thank you, Nathan. We get this question a lot. The next question, I think, Nathan, you're the best person to answer it. When will this be made commercial?
Okay, so the process is that we have to carry out these pilot projects on these protocols approved by the regulators. Excuse me. Then we take the data back to the regulators, and they carry out an assessment to look at the effectiveness that this would have as a mosquito control tool. And that assessment can take them 12 months, 18 months, that kind of time scale, once they actually have all the data in hand. And after that, they would then be in a position, if they think that this is an effective tool, to actually approve this for use. And that's the typical process that any new pesticide or biopesticide product like this mosquito has to follow in the US. So it's not possible to give a date for this. We obviously have to complete the field projects first, and then this goes back to the regulators.
Thank you, Nathan. The next question is related to that one, but it's for you to answer, Andrea. If it was commercial, would FKMCD buy it?
So at this point in time, I would say we're not ready for that decision. That's really why we're looking at doing these pilot projects to better understand, how it works. Does it work? And those are questions that, are ongoing currently. That's why we're doing these projects. But at the end of the day, this will be treated as we treat all of our other mosquito control products, right? We're going to look at how effective is it, and what does it cost? What is our, what labor do we need to support it? And see, does it even fit into our program? And those are all things that are going to be considered before we make any decisions to move forward. We just did an extensive cost-benefit analysis on new helicopters. We're going to be doing one on potential drone usage for us. So it's not something that we don't do on a normal basis. And it's definitely something we're going to do before making any sorts of decisions.
Thank you, Andrea. Nathan, this is going to be our last question. I'm sending it to you. It's, could I buy it, this is the Oxitec mosquitoes, as a homeowner in the Keys, or is it just for mosquito control districts?
Great question. So right now, it's not possible to buy this at all. This needs to go back to the regulators, and if the regulators approve this, then they would determine who would be allowed to buy this. Some pesticides are available to regular homeowners and to professionals, and that's definitely what we hope would be the case with this. And that is what we do in Brazil right now. So these mosquitoes are available for purchase online in Brazil. You can buy them, they'll be delivered to your house. You set them up, you add water, and a few days later, you have the male mosquitoes emerging to help control the local mosquito population around your house in Brazil. But in Brazil, it's also used by mosquito control authorities. So we have hybrid models of how this can actually be used. And I think in the future, we hope to have something similar in the United States. But that is still some way down the line.
Thank you, Nathan, and thank you both. Thank you, Andrea, for the really thorough answers. We're going to stop with that one. We're almost at time. But, if any of you have questions later, this florida@oxitec.com is monitored. We respond to everything that we receive there. We will post the recording from tonight. It'll probably be up tomorrow on our YouTube channel, on the keysmosquitoproject.com website. All of our past webinars are there and available for people to watch. And we just thank you all for your interest, and encourage you to stay involved and keep track of our project website, sign up for the listserv. We want to make sure you are receiving all of the information that we're sharing, and we really appreciate the interest. Thank you.